So today I'll be talking to you about our work on the discovery of natural products that potentiate current antifungal drugs. So I work at the National Center for Natural Products Research at the University of Mississippi, and we have an antifungal drug discovery program which has been funded by the NIH for the past 24 years. Alice Clark is the PI on this project, and I'm the co-PI. I've helped coordinate this project for the last 8 years. So in this program, we start with natural product extracts, and we screen them for antifungal activity. These extracts are crude extracts from a variety of sources, animal, plant, and microbial. Once we find hit extracts, we do bioassay-guided fractionation to isolate pure compounds. And then we characterize our compounds. We do medicinal chemistry to optimize our compounds, to improve their antifungal activity, and to reduce their cytotoxicity if that's necessary. And we also do mechanism of action studies. So our goal is to discover new antifungal drugs and new antifungal targets. So recently, we've also started looking for drug potentiating compounds. And here, what we're doing is we're going into our in-house repository of natural product compounds. We're not starting with extracts, but we plan to do that in the future. So we're starting with compounds in this case, and then we're examining them for drug potentiating activity with a focus on antifungal drugs. We are characterizing these compounds both at the in vivo level and at the mechanistic level. These in vivo studies are being done by Young-Sung Ban, who is a visiting professor in Joe Heitman's lab. So the work that I'm going to show you today was done by Sun and various people in Joe's lab. So again, the goal here is to find new drug potentiating compounds and new drug potentiating mechanisms and targets. So our strategy for identifying drug potentiating compounds is very simple. We make use of the classic checkerboard assay. And so this is a 64-point dose matrix assay. And we do both a microdilution assay in broth, and we also do recovery assays, as Leah had shown, where we take aliquots of cells and spot them on agar plates that lack the drug, and we look for fungicidal effects. The drugs that we are focusing on are amphotericin B, fluconazol, and caspofungin. And we are focusing on our in-house compounds. So far, we've tested 40 compounds, but our goal is to screen the entire collection of our in-house compounds. And I'll just tell you briefly that we have optimized and modified this assay where we have found that just a 16-point dose matrix, a 4x4 checkerboard, is sufficient to find good synergistic activities. So that's our plan for the future. So all the data that I'm going to show you today is work that was done on Cryptococcus neoformans, but we envision doing similar kind of work in Candida species also. So let me show you some examples of compounds that we have found. So here are two examples of amphotericin B potentiators. So this compound, NP129129, as you can see, is strongly synergistic with amphotericin B, has a good FIC, and it improves amphotericin B's MIC by 2, 4, 8, 16-fold. So this was very exciting when we saw this. And as you can see, this compound is inactive on its own. It doesn't have any antifungal activity. And the combination of the two is indeed fungicidal. This compound, NP129110, although it's not strongly synergistic, it actually improves amphotericin B's fungicidal activity by 8-fold. So to show you the structures of these compounds, NP129129 is actually an artemisinin analog. So this is an artemisinin dimer, and very strongly synergistic with amphotericin B. We've also looked at the selectivity index of our compounds. We have looked at their cytotoxicity profile in 3 different mammalian cell lines. And what we are comparing here is we're looking at the concentration of the compound that produces a fungicidal effect in combination with amphotericin B. So as you can see, it's 0.6 microgram per ml, whereas the cytotoxic activity is, IC50s are at least 10 times higher. So the selectivity index this is our assessment of selectivity index in this case, and it looks pretty good. NP129110 is a pyrrolopyrimidine. This was isolated from a marine sponge, and this compound, as you can see, is active on its own. Its selectivity index is quite good in 2 of the cell lines that we have tested. So here are 2 fluconazole potentiators, NP129121, very strongly synergistic with fluconazole, good FIC, and it improves fluconazole MIC by more than 20-fold. And as you can see, this compound is also inactive on its own. This compound also produces a fungicidal effect, and I should point out that these assays were done in a fluconazole resistant strain of Cryptococcus that we obtained from Joe. So in this strain, even at 50 microgram per ml, we never see a fungicidal effect. And of course, this is not a fungicidal drug, but as you can see, the fungicidal concentration of fluconazole over here is less than 1 microgram per ml. This compound, NP51365, not strongly synergistic, but it does produce a fungicidal effect in combination with fluconazole. So NP129121 is a synthetic peptide. The original compound was an antimicrobial peptide, and we have generated many analogs of that peptide. Its cytotoxicity profile is good. This compound, a peptide, should not be expected to be cytotoxic. NP51365 is an indolizidine. This was isolated from a plant, from the mesquite tree, honey mesquite tree actually. And this compound, it does have a few cytotoxicity issues, but this compound has a very interesting mechanism of action, and we have found that this compound actually inhibits cell wall synthesis. So we think that this is an interesting compound also. So here are 2 Caspofungin potentiators. As you can see, both of these are synergistic, good FIC values. They improve Caspofungin's MIC by several fold. And as you know, in Cryptococcus, Caspofungin is not very effective or is poorly effective. And both of these compounds produce a fungicidal effect. So NP75450 is a purine diterpene. This was isolated from a marine sponge, good selectivity index. NP75451 is a sesquiterpene quinone, also isolated from a marine sponge, and good selectivity index. Okay. So let me show you some in vivo studies that have been done here at Duke University. And these studies were done with our amphotericin B potentiating artemisinin analog. So the strategy that Sun used was to make use of the Galleria model, and we have seen other investigators use this model quite successfully for antifungal efficacy testing. And so what Sun did with the Galleria model is that he actually set up a mini checkerboard assay. So this is a 12-point dose matrix assay that he did with 2 different doses of amphotericin B and 3 different doses of NP129129, including all the controls and the vehicle. So these are 12 treatments that he did with 20 larvae per treatment. I mean, this type of an experiment would be almost impractical in a mouse model, So just another note about the drug injections. So Sun has modified this protocol, and he has found that he needs to do multiple drug injections to see a drug effect, and he needs to do early drug injections. So in this model, infection occurs early, and so drug injections also need to be done early as 4 hours post-infection. So out of all the combinations that he tested, there were 2 combinations that worked. And again, this goes to show that a model like Galeria would be really good for testing a variety of combinations and obtaining that appropriate optimal combination. So as you can see here, amphotericin B alone at 0.5 mg/kg did not improve insect survival compared to the vehicle. The compound alone also did not work, but the combination improved insect survival, and this is significant. The p-values are shown here. This is at 5 mg/kg of the compound, and we also found that a combination of same concentration or same dosage of amphotericin B and 10 mg/kg of the compound also improves animal survival. So using this information, Sun also did a mouse model study with this compound in combination with amphotericin B, and what he did here was he did basically 4 treatments. The vehicle, the compound alone, the drug alone, and the combination, 10 mice per group, and this is what would be practical for a combination study in the mouse model. Just a note about drug injections, again, multiple drug injections and early drug injections. So this is a tail vein injection model, and infection occurs early in this model. So in this case, as you can see, the compound alone did not improve survival. Amphotericin B at 1 mg/kg actually did improve survival, and the combination also showed improvement with a decent p-value. So we think this is a good starting point for us. We think we can improve upon this. We can easily see if we increase the concentration of the compound, or if we increase and do multiple dosing, we should be able to improve the efficacy even further. So I just wanted to emphasize that if we had not done the Galeria studies, this would have taken a lot of trial and error before we got to the right combination. So let me show you some mechanism of action studies that we've done on our drug potentiating compounds. And so far we've done transcript profiling studies, so that's the data I'm going to show you. So I just wanted to show you the strategy that we have used for our transcript profiling studies. And what we've done is we have done treatments where we treat cells with the solvent. In this case, I'm showing amphotericin B as an example. So the drug is at IC50 concentration. The test compound is at IC50. And the combination is also at IC50 of each compound, of the drug or the compound. And we played with this a lot. We tried different combinations, and it would have been nice if we could get the combination of the two producing 50% inhibition. That would have been ideal, but we could not achieve that reproducibly from experiment to experiment, especially in large-scale cultures. So we tried this idea. We just went with IC50 of each, and we tried a few compounds, and it worked really well. We were able to see nice drug-specific responses in our transcript profiles. So in our experiments, we exposed Saccharomyces cerevisiae cells to 1.5 doublings, and we found that this very early time point of just 4.5-hour exposure was sufficient to see drug-specific effects. We made use of Saccharomyces cerevisiae because we've done a lot of experiments with Saccharomyces with our in-house compounds. So we have quite a nice reference database of profiles in hand. So this would make data interpretation easy for us. And of course, gene annotations are more complete in Saccharomyces. So we did Affymetrix array experiments in this case. We plan to switch to RNA-seq in the future. We just bought Illumina and NextSeq equipment recently. But all the data I'll show you is with Affymetrix arrays. Okay, so here is the transcript profile of amphotericin B in combination with our artemisinin analog. So what we saw was amphotericin B alone causes the induction of a large number of genes involved in mostly stress response and also in transport and some other functions. And we have seen this profile before. This is a very typical profile of membrane stress. And we've seen this with amphotericin B before. What's interesting is that when you expose the cells to amphotericin B in the presence of our compound, these genes that were induced by amphotericin B alone are now not induced at all. They are not present at all. So what this means is that, you know, the adaptation response to membrane stress is being blocked by our compound. The compound alone produced a very interesting profile. It caused the induction of anaerobic genes. These are genes that are induced under low oxygen conditions, very typical set of genes here. And it down-regulated genes involved in respiration. And this combination of anaerobic gene induction and respiration gene repression is a very typical response of heme depletion. We have seen this before with some other compounds in our collection, which are actually, they inhibit heme biosynthesis. So we feel that this compound inhibits either heme biosynthesis or some aspect of heme metabolism. And this would be in keeping with what's at least speculated for artemisinin mechanisms. The mechanism is not very clear, but at least one of the mechanisms proposed for artemisinin compounds is that they interact with heme, and there's some evidence that they actually alkylate heme. And you know, this would also make sense in terms of the drug potentiating mechanism, since you know heme plays an important role in ergosterol metabolism. So one can imagine that you know messing up heme metabolism would eventually mess up the membrane. So the combination of amphotericin B and a heme depleter would cause even further membrane damage. So just to reiterate, our compound blocks the adaptation response to membrane stress that is exerted by amphotericin B. It most likely causes heme depletion by either heme synthesis inhibition or some other mechanism. And here's another thought. You know, this also suggests that heme depleters may act as amphotericin B potentiators. Okay. So here's the profile for a fluconazole potentiator. This compound is a piperidine alkaloid. This was actually isolated from fire ants. And I did not show you the data for this, but this compound also produces a fungicidal effect with fluconazole in Cryptococcus. So again, what we see is fluconazole alone causes the induction of a very typical set of genes which are involved in ergosterol synthesis and anaerobiosis. This is a very typical fluconazole response. And then what's interesting is that when you have fluconazole plus the test compound, these genes are either not induced at all or their induction is reduced. So again, that adaptation to ergosterol stress is being blocked. The compound itself generated a very complex profile. We saw induction of a variety of genes, and we saw repression of a variety of genes, and the combination of the two also induced a unique set of genes which were also involved in stress response. So at this point, we don't understand what the compound's mechanism of action is, but at least the combination appears to involve a block in the adaptation response to ergosterol stress. Okay. Here's a profile for a Caspofungin potentiator. This compound, NP75451, I showed you the data for this. This is a sesquiterpene quinone isolated from a marine sponge. This compound is called puhupihinone. This was isolated from a sponge that was found in Hawaii. That's why the name puhupihinone. So this compound, again, we can see the same theme. Caspofungin alone causes the induction of cell wall genes and cell wall maintenance and cell wall integrity genes. And these genes are not induced when you combine caspofungin and our compound. So again, blocking adaptation response. The compound itself causes the induction of genes involved in heat shock response and oxidative stress response. So same theme, a block in adaptation. So I'll tell you a little bit more about this compound and its mechanism of action. So we've done a lot of mechanistic studies on this compound, and we are finding that this compound behaves like a HSP90 inhibitor. So puhupihinone is structurally very similar to cilastrol, which is a known HSP90 inhibitor. And We know that it induces the heat shock response, at least from our transcript profile. We have done studies with the heat shock element LAG-Z promoter reporter, and it gets induced. We have done molecular modeling studies, and we actually do find that just like celastrol, bupropion interacts at the same amino acid residues that celastrol interacts with on HSP90. We have also looked at client proteins of HSP90. So we have looked at calcineurin, we have looked at SLT2 or MPK1, which is involved in the cell wall integrity pathway. And we've also looked at glucocorticoid receptor. All these are client proteins of HSP90, and our compound appears to inhibit their function. And with glucocorticoid receptor, this is actually a mammalian client protein, and there's a nice promoter reporters assay system that has been developed in yeast, where you can express the glucocorticoid receptor in yeast cells and then use a promoter reporter to detect its function. So in any case, all these studies have been done in Saccharomyces by us. Okay. So in terms of the mechanism of the combination, one thing that we looked at that Damien also showed this morning was to look at SLT2 or MPK1 activation. So as many of you know, Caspofungin activates SLT2, which is a key protein kinase in the cell wall integrity pathway, and SLT2 is a client protein of HSP90. So as you can see here, Caspofungin alone, if I show you the 60-minute time point, Caspofungin alone activates SLT2, but if you have Caspofungin plus bupropion, that activation is lost. Okay. So here's what we think is going on with this compound and with the drug potentiating effect. Caspofungin induces the cell wall integrity pathway, and RLM1 is the transcription factor that induces the cell wall repair genes. So our compound, bupropion, inhibits HSP90, and that in turn prevents activation of SLT2. So bupropion blocks the Caspofungin-mediated induction of the cell wall integrity pathway, and we are calling this it's a CWIP inhibitor is what we are calling it. So then we ask the question, can we find more natural products that work as CWIP inhibitors, and would these then synergize with Caspofungin? So we made use of a promoter reporter construct that Damien showed this morning. This construct was obtained from David Levin at Boston University. And so this basically has 2 copies of the RLM1 binding site hooked up to LAG-Z. And using this promoter reporter construct, we have developed a high-throughput screen where we take Saccharomyces cerevisiae cells, which contain the plasmid, we treat them with Caspofungin, and that induces LAG-Z, and then we look for compounds that decrease this induction. So it's a very simple assay. It can be done in a microplate format. And so as you can see, here's an image of an assay plate. The yellow color indicates beta-gal activity. And each well contains Caspofungin at the IC50 concentration, and each well has a unique test compound unique to each well. So, and column 12 has controls. As you can see, the clear wells indicate hits, which are CWIP inhibitors. So you can see several hits here in column 11, B11, C11, D11. And as it turns out, all the hits that we found on this plate were analogs of the same parent compound. So we do a primary screen at 1 concentration of the test compound, a secondary screen at 3 concentrations, and our tertiary screen is a checkerboard assay with Caspofungin. And so far we have screened about 4,000 compounds from our in-house collection, and we have found 126 hits in the primary screen. I know this is a high hit rate, but we do have a lot of analogs in our collection. And our chemists have synthesized analogs over time for SAR studies or for modifying the activity of our compounds. So just to show you, we have a variety of different structures that we have found. They belong to different structural classes. Some of these are very simple, very amenable to synthesis. And what's nice is that the 2 compounds I've highlighted here are NP75451, this is bupropion, and NP75450, this is the purine diterpene that I showed you earlier. Both of these are synergistic with Caspofungin, so that's nice to see that we found these hits in this screen also. So our conclusions, as you all know, in vitro studies are showing that, synergistic compounds or drug potentiating compounds can decrease the effective dose of the drug. They can convert a fungistatic to a fungicidal, and they can also, improve the spectrum of activity of a drug. Our in vivo studies are telling us that Galeria is a good model for studying drug combinations, and our mechanistic studies are telling us that our compounds seem to be blocking the adaptation response to the stress exerted by the drugs. And we feel that identifying these drug potentiating mechanisms can help us identify even new drug potentiating compounds. So just to acknowledge everybody, Siddharth Tripathi is a postdoc in my group who did the majority of the work, and Chin Feng is the research technician who helped him. Xingkong Li is the chemist in our program. He isolated many of the compounds I showed you. Melissa Jacob is our compound repository manager. Alice Clark is the PI. And many thanks to Sun and everybody in Joe's group who helped with the in vivo studies. And David Levin gave us the plasmid. And thank you for your attention.